biology revision video with Mr. Phillips, and today we are looking at epigenetic control in eukaryotes, and this is part 2 of the video, which is going to be focusing on what environmental factors can affect the epigenome and how these things are controlled. So hence, let's keep moving forward then. So just to remind you some of the areas we've talked about previously, there are something called transcriptional or transcription factors, and all cells we know have the same genome, but they have to function differently. So some genes are expressed in different cells and some genes are turned off in different cells, but all cells have the same type of housekeeping genes. And we've talked about transcription factors in the previous video, where these attach to the promoter region of the genes that need to be activated. That allows RNA polymerase to attach, and that allows transcription to begin. And please go back and watch that video if you do not know about that. About epigenetics in the last video. So we said originally we believe that your phenotype could not be passed on and inherited changes to your phenotype due to the environment. But we now believe that there are environmental factors that can cause heritable changes in the gene function without altering your DNA sequence, and that is called epigenetics. And it looks at how the environment can influence your genetic inheritance as part of your offspring without altering the DNA sequence. At what this means, and basically it's a change in gene function without altering the sequence. And we linked it to twins, and we said the actual structure of DNA is DNA wrapped around the histone protein. Remember, there are 8 histone proteins normally in one epigenome, and then there are something called chemical tags that cover the DNA, altering how tightly it is wrapped around them. And the chemical tags form this second layer known as the epigenome. And the epigenome therefore determines the shape of the DNA and histone. Important is because how tightly that DNA is wrapped around the histone complex determines whether that gene is switched on or switched off. So if it's tightly wrapped around the histone complex, the DNA can't be transcripted, and therefore the gene is turned off. And if it's loosely wrapped around, it can have transcription, and therefore the gene is turned off. Got to last time. So those chemical tags can be therefore altered by environmental factors impacting. So the chemical tags can attach the DNA and the histone, altering how tightly it's wrapped around. If the DNA, as I said, is wrapped around tightly, then the gene is turned off as they cannot be read, and it's called epigenetic silencing. If the DNA is loosely wrapped around, it's turned on, and it allows the bases to be exposed, allowing transcription. So it allows the transcription factor to attach, it allows the RNA polymerase to attach. Now, the chemical tags are not fixed like DNA and can change the environment, resulting in DNA wrapping tighter or looser over. Key bit here then, so chemical tags respond to the environmental factors altering the epigenome over a lifetime, causing them to inhibit or activate specific sets of genes. And environmental factors can signal and cause the changes to those tags. And basically, the environmental factors might cause specific proteins to enter the cell, and that might cause other proteins then to interfere, and this message gets passed through other proteins to the nucleus where it has this impact. And basically the message passes through specific proteins, which can then attach to specific DNA bases, having one. The effects that we're going to be looking at is which can alter the expression of your DNA is methylation, which is when you attract enzymes can add or remove methyl groups, and acetylation of the histone, and again we'll talk about that. So you've got two different things here, methylation and how that can alter the expression of the DNA, and acetylation, which is first of all. This is when methyl groups are added to the DNA, and normally they attach to the carbon in the cysteine cytosine organic base. Now, when methylation occurs, this normally suppresses transcription and it therefore turns off the gene. This therefore, how does it do it? Well, it prevents the binding of a transcriptional factor binding to the promoter region, which or it can attract proteins which condense the DNA histone complex, making it tighter. So if you have a chemical which increases the amount of methyl groups added to the DNA, this can suppress transcription, turning the genes off. The gene is not being exposed, or it prevents the transcription happening. The other way we talked about is acetylation of histones. So now we're talking directly about the histone rather than the DNA structure itself. Okay, and histone is made up of lysine, is the main amino acid in histone, and acetyl groups can be attached to this. Often acetyl groups come from coenzyme A, which you will have heard of from respiration, and it will attach to the histone, replacing a positive H plus ion, making it much more negative. And this removal of the H plus ion makes the DNA wrap less tightly then around the histone. So the genes become activated, they become turned on. Now, RNA polymerase then can easily attach, and this will increase the transcription of the gene. So if you have what we call hyper acetylation, then that is adding acetyl groups, and that will stimulate transcription. If we do deacetylation, which is when we remove acetyl groups, meaning we're adding H plus onto the histone, which means it can then closely associate with the DNA, and the DNA wraps more tightly around it. So if we remove acetyl groups, this inhibits transcription. So in terms of epigenomes and epigenetics, if there are certain environmental factors that will add acetyl groups, that will cause the gene to be turned on. And if there are certain environmental factors that remove acetyl groups, that will turn the gene off, inhibiting transcription. Effectively, but is exactly as examples of where we know epigenetics has been studied. So in rats, basically we can see that offspring that have good care when they are young will end up being good nurturing parents when they're older. So if, for example, if a pup is raised by an anxious parent, that pup becomes anxious when it's older, and if it's raised by a highly nurtured parent, it will become a highly nurtured parent in the future. In humans, an example of gestational pregnancy, which is when if the pregnancy, if the female, if the mother gets like diabetes during pregnancy, that baby will be exposed to higher glucose concentrations. If that is a daughter, that daughter's DNA may change as a result, and then they are more likely to develop gestational diabetes themselves when they are pregnant in the future. Genetics can be inherited, and that is a key thing. So these changes to the chemical tags, which can make the histone wrap tighter or looser, can then be directly carried to the next generation. Because we inherit both genes from the parents, and basically there are mechanisms to try and stop this. So during early stages of development, specialized cellular mechanisms will search and try and erase epigenetic tags to return the genetics to a clean state, or to a clean slate, I should say. However, some escape being erased and then pass unchanged from the parent to the is a condition called Prader-Willi syndrome. And the symptoms of this disease is constant hunger, which leads to obesity, restricted growth, reduced muscle tone, no development of secondary sexual characteristics, and learning difficulties. Now, this is a complex disease because it involves the inactivation of genes and it involves missing genes. So normally what happens, so this is in a healthy individual, the genes on chromosome 15 from the mother are inactivated. This is 7 of the genes are inactivated through epigenetic inheritance, meaning most people only have the genes expressed from the father on the paternal chromosome, and that is the normal healthy inheritance. However, if there is a mutation, what happens for people who end up with this disease is that the father's 7 genes are actually deleted due to mutation. So you only end up with 1 set of genes, but these have been silenced on the mother's chromosome, and therefore you don't have those working functional genes in your body. So epigenetics, obviously, and we'll do in the next video on this, are links to potentially cancer and tumours, and it has lots of importance in cancer research. So tumour DNA shows modifications in methylation and the histone protein. So when we look at this in a bit more detail, DNA can be mutated, the methylation can cause DNA to switch on certain genes or switch off certain genes, and that can therefore affect the risk of cancer. And drug development as a result is looking at how we can remove acetyl groups or methyl groups to cancerous DNA. And again, we'll look at this. So let's just have a quick look at it now, and there's another video coming up on this. Mitosis is controlled by 2 main types of genes. There is something called a proto-oncogene, which controls cell division, and then there is something called tumor suppressant genes, which slow cell division and stimulate programmed cellular death, called apoptosis. Now, apoptosis is not a bad thing. Apoptosis is when your cells are programmed to die after a certain number of divisions, called the Hayflick's constant, and that then means you don't have too many cells in your body. So basically, the proto-oncogenes are ones that control cell division and only make it enough cell division. So mutations can occur, turning proto-oncogenes to oncogenes, and they lead to uncontrolled cellular division. Now, they code for growth factors that stimulate cell division by binding to protein receptors to the cell membrane. Oncogenes may produce too many growth factors or start triggering division in cells without receptors, and we'll look at that more in the next video. Tumor suppressant genes slow the cell division down normally and stimulate apoptosis, but if they become hyper methylated, then what will happen is that will then cause the DNA to wrap around the histone tighter, as we said earlier, and those genes may become inactivated. And if the tumor suppressant genes become inactivated, then this can cause cell division that is which in both eukaryotics and prokaryotics, translation can obviously is involved in producing proteins, and the gene can be inhibited from working and producing those proteins by breaking down the messenger RNA before it becomes coded in the ribosomes to make the polypeptide. And basically, a small type of messenger of RNA, called small interfering RNA is used to break down the messenger RNA before it can be used. So let's have a look at how this works. So how this works is we start off with a double-stranded section of RNA, which is going to be complementary to the bit that is going to be transcribed, or has been transcribed and will be translated if nothing else happens. And this double-stranded RNA is broken down by an enzyme and it's probably a restriction endonuclease, into a small interfering RNA molecule, or siRNA. Now, this small interfering molecule then gets split into individual strands, and the single strand of this attaches to an enzyme, as you can see here. Now, what that does then is those single strands will then pair with the complementary bases on the messenger RNA that is about to be translated. So you can see here that's coming in, that would be going to the ribosomes, but instead it attaches to the exposed bases from the double-stranded DNA you have cut up previously. And then that enzyme cuts the messenger RNA that would be used to translate into different bits. This means that messenger RNA has now been cut into smaller sections, and it prevents translation from happening and can't be translated into a polypeptide. This means that gene has been blocked as a result. And obviously this is a complex process, but this is just showing you how we can further block the expression of a gene using a different mechanism, which is called messenger RNA silencing, using small interfering RNA, which basically uses an enzyme to cut the messenger RNA into smaller pieces, so it has been transcribed, but it can't be translated at the ribosome.